Last class we covered the topic of synapses. So that was 8A3. We talked about how action potentials travel across the nerve fibers and then how they move from one neuron to the other across synapses using neurotransmitters. Then we talked about how drugs have an effect on the nervous system, right? And then today, staying on topic, this is chapter 8A5, and it's titled Sensory Systems and Detection of Light. So as the name suggests, we are going to talk about, right, how we sense stimuli, and in particular, we're going to talk about the detection of light. So we're going to talk about the eye, right? But we're not going to talk about the eye as in, its anatomy and physiology in this chapter, although there's a very detailed illustration given. We're going to focus on, the sensory receptors in the eye. So, right, for a living organism, right, the point of the nervous system is to coordinate everything that's going on and to respond, right, what's happening outside the body, what's happening inside the body, and to try and maintain life, right, by producing appropriate responses. And so sensory receptors are very important, right, because they gather information about the internal and external environment. Let's write that down. And obviously external environment, right? And obviously this ties into the topic of homeostasis that we've done earlier. Now, right, there are two types of sensory receptors. One of them are neurons and the others are specialized cells. So, right, a primary receptor, right, is a neuron. And right, it has a dendrite that is sensitive to one particular stimulus. And so when it detects that stimulus, or when it receives that stimulus, the whole, events of an action potential going off and all that happens. Then you've got what are called secondary receptors. Right, and these are more specialized cells and, right, not neurons. and right, their own structure and their mode of function is different and rather more complicated than the primary neurons, the primary receptors, right? And they're also sensitive to a particular type of stimulus, right? So it's not like these guys are work in different departments. No, they are specialized. Okay, and right, so there's a synapse between the regular sensory neurons and right these guys, and they carry an the impulse to the central nervous system. What they're calling normal sensory neurons, and at the end they take the impulse or the signal or the stimulus towards the central nervous system where it's processed, and then whatever response is required, right, is taken by the motor neurons, or if it's just happening in the brain, right, because some things just end up in the brain to be processed. Okay, so there are a number of different types of receptors, right? The more complex the organism, the more things, right, the more specialized it is, and higher order systems and sense organs are what you would expect. And so, right, let's just go to the next page. So types of receptors. So there's what's called the proprioceptors, and these have to do with they're sensitive to the position of the skeleton and muscle contraction. And so they are involved in, for example, the posture of our bodies, right? Then you've got chemoreceptors. And right, obviously they're sensitive, as the term is, to chemical stimuli. And examples of chemical stimuli are smell, taste, and pH. Then there are mechanoreceptors, the mechanical stimuli. And those would include pressure.
Sorry, sir, I accidentally left.
I didn't even notice this. I'm sorry. I was just going on about this.
No, I just left like a second ago,
Okay.
accidentally.
So pressure, tension, right, movement, and gravity. Then you've got photoreceptors, which are what we'll be talking about, because we're going to talk about the eye, and these are sensitive to stimuli from the electromagnetic spectrum. Right now, in humans, it's visible light. Right, but some animals are also able to detect UV. Right, or even infrared. Some animals can see infrared. So, and then finally, you've got thermoreceptors. And as the name suggests, they are sensitive to changes in temperature. Okay. So how do sensory receptors work? What happens is basically what we've talked about in terms of action potential firing. There's just a slight difference, right? So inside the receptor cells, right, are negative on the inside, just like a nerves or a neuron, a nerve fiber or a neuron. And when they receive a stimulus, right, sodium starts moving in. And it goes positive, right? The potential difference changes, right? So there's obviously a chemical, electrochemical and concentration gradient for sodium. It's moving in rapidly, and right, instead of being called an action potential because these are receptor cells, it's called a generator potential. Right? So the stimulus sets up generator potential. And right, if it's a small stimulus, then the generator potential will be small. If it's a large stimulus, right? So if it's a large stimulus, you're going to have a large generator potential. And if it's a small stimulus, you're going to have a small generator potential. And by saying that, right, at the receptors, right, these do not follow the all or nothing. So in regular nerve fibers, it was all or nothing. So there was a threshold. And if the depolarization reached the threshold, then the action potential would definitely fire. And if it did not reach the threshold, even if it was close, it wouldn't fire. So it either happens or it doesn't happen. That's not what's happening at the receptor cells. A small stimulus will generate a small generator potential. A large stimulus will generate a large generator potential, right? So over here, it's not being followed. However, right, this generator potential is passed on to the the sensory neuron, right? So what we're talking about is the receptors, right? So this is passed on right? And over here, right, an action potential is only generated if it reaches the threshold. Because it's a neuron, it's going to follow the all or nothing, right? So just remember that difference. And the receptor cell, it's not going to, be for, so again, small gives small potential, large stimulus, large potential. Whatever's transferred onto the sensory neuron, there, because it's a neuron, the threshold needs to be met for an action potential to be generated, right? Now, in that case, right, what happens basically is, right, you have several receptor cells that are forming synapses with a single sensory neuron. And how does that help? That helps because, right, if the generator potential from a single receptor cell is not, large enough for the threshold to be reached and the action potential to be put into, set up an action potential in the what you call it, the neuron. But if you've got, for example, 3, 4, 5 different receptor cells forming a synapse with the same sensory neuron, and so you've got a small generator potential coming in from 5 different receptors. And so individually they're small, but they're all going to converge, right? And then together they're going to help the depolarization in the sensory neuron to reach the threshold and therefore an action potential to be generated, right? And so, right, the generator potentials have a combined effect in setting up the action potential, right? And this is called convergence, right? So a term you want to keep in mind. Okay. From here, we move on to talking about uh, right, the human eye. And so, right, the eye is obviously sensitive to light. It is a very sophisticated sensory organ, and it's able to detect electromagnetic radiation in the wavelength of 400 to 700 nanometers, which is basically the range of visible light, right? Uh, there's a diagram uh, given. I'll just show it to you. Now, right, all of this detailed diagram, we are not talking about this. The only thing we want to talk about is the retina, which is this entire layer that's given the green color. And this is where the photoreceptors are in the eye. And we're going to mention the fovea. right, which has the highest which right has only one of the two types of photoreceptors called cones, right? The other ones are called rods. And then the blind spot is where the optic nerves all meet, so and then leave as one bundle, right? So all the neurons gather over here and they form the optic nerve and they leave. And so this part, the blind spot, has no photoreceptors. So when we say, oh, I didn't notice it was in my blind spot, that's basically meaning that the angle at which the image was being formed, right? Even though perhaps the eye was picking up in terms of the light coming from the object and being moving through the cornea and everything and the lens and then coming in, right? So maybe that was happening, but the region where that image was technically forming was the blind spot, and since there are no photoreceptors, it wasn't being processed, and right, so that's what's happening. Now we're not interested in everything that's happening over here, right? So what we're going to talk about is basically the retina. So let's go back to the whiteboard for that. Okay. So whatever light enters the eye is focused on the retina. Right? And the retina contains, right, the receptors, photoreceptors, in fact, let me write that. Right, which are called rods and cones. The majority of the ones we have are rods, and comparatively there are only a few cones, right? So what they mention, this figures, there are about 100 million of these photoreceptors in each eye, right, in the retina of each eye. And right, about 90 million or so are rods, and only about 6 million of those are cones. Now what's the difference between these two? So the rods are, right, contain a pigment called rhodopsin. Rhodopsin, we'll talk about that later, right? And they are sensitive to low light intensities. So they respond to low light intensities. And with that, right, they give us black and white vision and are also sensitive to movement. So they give black and white vision. So they got nothing to do with the color that we see, right? And sensitive to movement. So if you're picking up anything that's moving, like you're seeing my cursor moving across the screen while I'm scribbling, right? Your rods are active. Then the other ones are cones. And right, they are mostly concentrated in the region called the fovea, although not in, like, it's not like they're found only in the fovea, right? So the out of the very low number that there are in the retina, out of 100 million, just about 6, right? The majority of those are in the fovea. And what these guys do is they contain the pigment, right? So the fovea is part of the retina, just to clarify. And they contain the visual pigment. This is also visual pigment called iodopsin. And they are not sensitive to low light intensities, they are sensitive to higher light intensities, meaning they respond to brighter light. So they're responding to bright light. And what do? They give us color vision. And right, clarity. All right, so they give us clarity of vision. And just to add it over here, the fovea, because it has a concentration of the cones which give us clarity of vision and color, what that does is, right, it gives what is called great visual acuity. So acuity, right, basically means the ability to see clearly. It actually means the ability to think, hear, or see clearly. But since you're talking about visual acuity, basically this term visual acuity means, right, ability to see clearly, which just ties into the function of the cones, right? Clarity of vision. So the more cones there, because they're concentrated in the region called the fovea, right, we get, right, very clear images, especially if they're being focused directly onto the fovea. Okay, so what do we have happening? Right now, these are picking up light, and that light is being, that action potential that's generated in the sensory neurons and taken to the central nervous system. What happens is that, right, the impulse reaching the CNS is processed by the brain. Right, and what it sees is, oh, first of all, it's coming in from photoreceptors, so it has something to do with light and color, and plus which ones. Right, and so the brain processes that as an image. So in terms of action potential, what we are hearing, smelling, tasting, touching, seeing, right, the five senses, in terms of action potential, they're all the same. So how does the brain distinguish the color of a food item that I'm looking at, and the smell, and the taste, and the texture, right? And if it's like boiling or sizzling, or it's making some sort of sound, for example, like those Chinese sizzler plates, and then so the sense of hearing as well. And so I'm receiving those five stimuli, or if it's a fizzy drink, then it goes like that fizzing sound. Anyway, so I'm receiving five different stimuli from the same object. And so what the brain does is, even though in terms of action potential what's reaching the brain is the same, but how it's processed is which receptors is it receiving it from, right? That is what makes the difference. And from which region of the body. So if the receptors are coming in from the nose, then okay, so those are chemoreceptors from the nose, so it translates that to a smell, whereas the chemoreceptors in your tongue translate to a taste, and the chemoreceptors in your ears translate to, for example, well, no, those are not chemoreceptors, those are other kinds of receptors, but right. uh, so right, when you feel that it's hot, right, so those are thermoreceptors in your skin, and right, obviously the photoreceptors, the item that you're seeing. So that's how it works. Now, the structure of the rods and the cones are are illustrated in the book, and they've got. I'll show it in fact instead of just going over it verbally. Do we have time? Uh, will it be okay if I stop here?
Sure, sir. No problem.
Even though there's not a lot left, I actually have to uh, do something after this, and so I don't want to leave this explanation midway. All right, so I'll stop here. This is right, I guess, a reasonable point, and then we'll continue next class.
Sure, sir.
All right. So I'll see you next time, inshallah. Any questions, by the way? Is everything clear?
Yes, sir.
Okay.
Alhamdulillah.
All right.